Okay, good afternoon, everyone. Welcome to this wonderful spring day in Boston. And our speaker today tells me it's a wonderful spring day in Ireland as well. So I'm particularly delighted to be able to introduce one of my longstanding colleagues, Professor Mary Ward, who is a professor of nutrition and dietetics at University of Ulster in Northern Ireland. And Mary works at the Northern Ireland Center for Food and Nutrition and Health, and is focused, like I have been, on B vitamin and one carbon metabolism for some years. Mary and her group have done wonderful studies, both observational clinical studies as well as several very promising and insightful intervention trials. And she'll be discussing a lot of those today. She takes advantage of several large cohorts, such as the Irish National Adult Nutrition Survey and the TUDA study, which I believe stands for Trinity Ulster Department of Agriculture. Is that it, Mary? Okay. And the Genovit cohort as well. So as I mentioned, Mary's been involved with one carbon metabolism for some years. So we run into each other on a regular basis at meetings. And most recently, her group has been unveiling this very interesting kind of intersection between the MTHFR common polymorphism, riboflavin, and blood pressure. And Mary, I certainly hope you're going to be talking about that or including that in your talk today. Mary publishes regularly in top-ranked journals across several disciplines. She's been quite successful in securing external funding from a number of government agencies in the UK and Northern Ireland and the EU, so on and so on and so on and so forth. And let's see, she's currently Deputy Director of the Wellcome Trust Wolfson Northern Ireland Clinical Research Facility, and is current President of the Nutrition Society in the UK and Ireland. So I'm not going to take up any more of your time, Mary, and I'm going to let you go. But before I do so, I just want to remind people to put their questions in the Q&A icon, not in the chat. The chat's going to be disabled, and we'll have a few minutes at the end for any questions that you might have for Mary. Okay, Mary, take it away.
Thanks very much, Joel, and thanks for that lovely warm introduction. I'd also like to thank Eleanor and the group at Tufts for inviting me along today and giving me the opportunity to talk about the work that we've been doing, as Joel has said, for very many years now. So I wouldn't like to count, but it's lovely to be here. Okay, so I'm going to talk to you about B vitamins. Oh, if I can get my cursor to change. But firstly, just to give you a moment to show you where I am. As Joel said, I'm based at Ulster University, and we have 3 campuses right across Northern Ireland, and we're located here in the middle, just in the middle of the north coast of Ireland, just a stone's throw from the world famous Giant's Causeway, if any of you have had the opportunity to come, and one of the oldest distilleries in Ireland, Bushmills, which makes the famous Bushmills whiskey, and also some of the scenes for Game of Thrones, if anybody has watched that. And this is Mussenden Temple, and you can see it in the distance. This was a photograph I took on Saturday while I was out for a walk, and you see the sun does shine in Ireland sometimes. Okay, so I'm based at the Nutrition Innovation Centre for Food and Health, and our director is Professor Helene McNulty, a close colleague of mine, and really we've worked together for very many years in this whole area. And we're a group of about 50 to 60 at any one time, academics, postdoctoral researchers, PhD students. We've also we're also responsible for teaching undergrad nutrition both nutrition and dietetics at undergrad and postgraduate level. So we're a busy centre, and our main aim here is really in preventative, so how nutrition plays a role in the prevention of very many chronic diseases. And so across niche, our group, we have people who are interested in cardiovascular, bone health, gut health, mental health, obesity, and that's from early life right through to nutrition and aging. And I'm part of the folate and B vitamin group, where Helene and I lead a group, an active group of researchers, and we're going to present the work that we have done over, quite some years, and bring you right up to the very present time. So I'll be focusing a little bit on riboflavin. This is one of our favorite B vitamins, and somewhat overlooked, and I'm really going to probably highlight some of the reasons as to why this is an overlooked nutrient. I'll go on then to look at some of the metabolic interactions with riboflavin and consider some of the metabolically related B vitamins, and really with a consideration of where they're playing a role in optimizing health right across the life cycle. And then before I finish, I'd just like to introduce you to our new co-Centre for Sustainable Food. I am co-leading the nutrition and health platform of this 32 million initiative that's happening across the island of Ireland. And so I just thought it would be interesting to close the talk with some mention of that. For the most part of today, I will be centering around B vitamins and that are metabolically linked through one carbon metabolism. And as Joel said, that's what's given us the opportunity to meet on very many occasions over the last 25 years or so. But just to highlight for those who might not be familiar with this metabolic pathway, a number of B vitamins play a key role in one carbon metabolism. So here on carbon metabolism, where we have the generation of folate, which is a really important molecule that plays a pivotal role in generating methyl groups for the generation of SAM, the universal methyl donor, and so therefore, in terms of metabolic control, folate is a really important nutrient. And in the folate recycling pathway, a number of B vitamins play a role. So we can see here that we've got vitamin B6 playing a role here in the conversion of one of the folate cofactor forms, tetrahydrofolate, into 5,10-methylene THF, but also in the transsulfuration pathway, where we're degrading homocysteine. And again, vitamin B6 plays a role here. In the generation of 5-methyl THF, which is the main form of folate that really, I suppose, takes part in a lot of metabolic reactions, the enzyme MTHFR is a really important enzyme, and I will be talking about this enzyme a little bit more. But for now, I just want you to remember that in order for MTHFR to work effectively, it requires riboflavin in the form of FAD as a cofactor. And a common polymorphism exists in this enzyme, which about 10 to 12% of the global population have, but which it has a frequency much higher in some countries. So for example, if you were born in Mexico, 1 in 3 people might carry this polymorphism. I understand it's even higher in other Central American countries such as Guatemala. And in northern China, there is also a frequency or a prevalence of about 1 in 5 people carry this polymorphism. So it's quite a common polymorphism. Vitamin B12 is also really important in this remethylation pathway here. So it's the remethylation of homocysteine to methionine. Vitamin B12 plays an important role there. So in terms of what I'm going to present to you today, you'll see that I talk about B vitamins in relation to a number of chronic diseases. And what I think is really happening here is that When we dysregulate this one carbon pathway, when one of these B vitamins are low, then one carbon metabolism is affected, and that can then lead to suboptimal health through a number of different mechanisms. So it's likely that methylation has an important part to play in that. But sometimes people, query as to how can B vitamins be involved in all of these chronic diseases. And really, I think it's the disruption of this pathway and affecting the balance of these B vitamins. So to start with then, I'm going to start with riboflavin, as I mentioned, and I'm not going to give you a biochemistry lecture, but riboflavin has a fundamental role in redox reactions and in energy production. But for the purposes of today, I'm going to focus in on its role in one-carbon metabolism, as well as its metabolic interaction with vitamin B6, and this is outside of one-carbon metabolism. So riboflavin actually has coenzymes. It has two main coenzymes, FAD and FMN, and they take part in about over 100 metabolic reactions. So it really has a ubiquitous role. It's, in terms of its sources, it's widely distributed in small amounts, but most important dietary sources are milk and dairy products. So it's a very important nutrient, but because it's widely distributed in foods and because it's involved in so many metabolic reactions, we sometimes don't see the effects of suboptimal riboflavin status. There are no main clinical signs, if you like. From a deficiency point of view, deficiency is common in low and middle income countries, and in terms of the signs of deficiency that you would expect to see, I've lost my cursor, would be chelosis. So you've got this drying and cracking of the lips, or you've got a shiny tongue. And these would be characteristic of riboflavin deficiency, but also are shared with some other micronutrient deficiencies. So it's sometimes difficult to diagnose. And for that reason, we have been sent samples from some low-income countries where they're trying to establish whether or not it is riboflavin deficiency, because we're one of the few centers globally that actually measure status on a routine basis. So what's probably less well studied or less well talked about is a biochemical riboflavin deficiency. And we estimate, this is important because we estimate that about 40 to 50% of reproductive aged women, but also men, are affected by biochemical riboflavin deficiency. And this occurs even in high-income countries where we have what appears to be adequate riboflavin intake. There are no visible signs of riboflavin deficiency, but it is associated with disturbances in one-carbon metabolism, as I'll go on to show you. And I'll also talk about the work that we have conducted in relation to hypertension and anemia. So why should we be worried about riboflavin? Well, in a very nice paper published very recently in the Lancet just last year, in Lancet Global Health, where micronutrient intakes globally were reported. And this was a really nice paper because data was taken from all of the individual countries for this modeling exercise. And so this is probably about the best available data that we have in order to report inadequacy. And the conclusion by Simone Passarelli and the authors was that in terms of riboflavin, about 55% of the global population have inadequate riboflavin intake. And here we can see in this very clear map that obviously the countries that are moving towards red, or for the kind of yellow or red part of the map, you can see that the situation is much worse. Whereas the countries that are green and blue, riboflavin inadequacy is not a major problem. What I would say, however, is to remind you that this important paper highlights inadequacy, but it's based on dietary intake data, and we do not have an equivalent paper that reports biomarker status because largely we do not have biomarker data. So for example, in the US, you have no national biomarker data for riboflavin status. So really what I'm saying is that this is probably the best case scenario, that really if we had access to better data, that we might see more inadequacy than we're generally familiar with. And just to give you a little bit of an indication into intake and status from a global perspective, and this is a very brief summary because I haven't time to go into it in any great detail, but globally our dietary reference values are similar enough across the world. I'm focusing in initially on the UK and Ireland because these two countries do have national riboflavin biomarker status reported. In the UK, it's on a rolling basis, and in Ireland, on a less frequent basis, but really as and when the money arises to do a new population measurement. So we do have biomarker data for these two countries. And what we see is that the DRVs in Ireland, they're based on EFSA, so the European Food Safety Authority, are probably they're the group that have most recently revisited riboflavin DRVs and have set them a little bit higher than they are in the UK, the US, and in China. It's about 1.4. So it's largely ranges from about 1.1 to 1.6 milligrams per day. And if we look at the best estimates we have of dietary intake data, we can see that largely populations are meeting these intakes. In the UK and Ireland, intakes are a little bit lower than the US, and this is explained by the fact that in the US and Canada, there is an enrichment program which adds a certain amount of riboflavin back into flour that has been lost in milling. So this 3.9 milligrams in the US also reflects supplement users. And I took that data, that was data from a 2024 paper that was published in Frontiers in Nutrition. I also found data in the US for older adults, which was in a more recent paper that was published just last year as well. And this is again in Haynes, which estimates that in older adults, the average intake is about 2 milligrams. But clearly there will be groups in the population who have lower intakes than that. The picture in China is much worse. Intakes, the average intake is much lower, and that's because in China, the main sources of riboflavin in the diet are plant-derived and so are much lower. And the reason why we don't have good biomarker status of riboflavin is that our gold standard measurement is currently considered to be a functional assay. It measures an enzyme, glutathione reductase in red cells, erythrocyte glutathione reductase, and it's an activation coefficient. So it's a functional assay. It's measuring enzyme activity in the presence or absence of FAD and calculates a ratio. That's considered the gold standard, but the method requires thrice washing of red cells, so it's by no means a convenient measure out in the field. We also can measure urinary riboflavin, and I note from some of the early papers from the US that have reported riboflavin status that they were largely based on urinary riboflavin, but again, a very inaccessible marker. And so in terms of accessible markers, serum and plasma would certainly be much more accessible, but we don't have very robust data on serum or plasma markers of riboflavin. And so the data that are published on the population from the UK and Ireland are based on the HNRCA assay. And this is just a summary of the HNRCA assay. And here we have the cutoff points. So if you have an activation coefficient of less than 1.3, you're considered optimal. 1.3 to 1.4 suboptimal, and greater than 1.4 is deficient. So the higher your HNRCA value, because it's an indirect or a functional marker, the worse your status. So it's important to remember that. We received funding from JPI. It's a European initiative to look at this whole problem, and we teamed up with Yvonne Lammers, who's some of you might know, and she's based at UBC, and also colleagues in the south of Ireland led by Professor Albert Flynn. And what we really wanted to do in this project, which we named the DERIVE project, was to try and develop or validate easily accessible riboflavin markers and demonstrate important functional, gene, nutrient, and health effects of optimal riboflavin status. So that was the DERIVE project. And what we've shown, we haven't published this paper, it's in preparation, but what we showed, we looked at the three main cofactor forms, riboflavin, FAD, and FMN, and we showed that of these three forms, these three cofactor forms, plasma riboflavin certainly shows the most promising. Here, what we did was we looked in the NANS population. This is the National Adult Nutrition Survey in Ireland, and this is a representative sample of the Irish population. And we divided the cohort into those who were deemed deficient, suboptimal, and optimal, and measured plasma riboflavin, FAD, and FMN. And although plasma FAD and FMN were not good at determining deficiency versus sufficiency, plasma riboflavin was able to do that. And in fact, when we looked at randomized control data, we were also able to show that plasma riboflavin was in fact a robust marker. So we hope to publish that paper, and that will, I suppose, give us a much more accessible biomarker that will help us to get much better global data on riboflavin status. So watch this space for that. Okay, so just to give you a little bit more data from the DERIVE study, what we showed here was, so this is the National Adult Nutrition Survey in Ireland. And in fact, when you measure riboflavin status, what we see is that quite a significant percentage of the population appear to have riboflavin deficiency. So, appear to have riboflavin deficiency, which is this gray part at the graph. So greater than 1.4 representing deficiency. But of interest, what we showed in the DERIVE study, because we had access to the BC Generation Project, so that was 1,200 adults who were living in the Vancouver area of British Columbia. While they weren't entirely representative, we aimed to be as representative as we could in terms of males, females, but also different ethnic minorities that lived in that area. And we demonstrated that about 24% of that population had deficient riboflavin status. And that's despite the fact that there was an enrichment program in Canada. So we really need much better data. We've looked at populations around the world, and we have shown that in both low and high-income countries that there is probably more riboflavin deficiency than people generally recognize. So here we see the white area of these graphs shows Ireland and the UK, very similar in terms of our ERAC, of where our level of deficiency as determined by ERAC, a little bit lower in Canada, but nonetheless still there despite enrichment. And then in stark contrast to the low income countries where there is a very high prevalence of riboflavin deficiency. And all of this data was generated in our lab. So we have some degree of confidence looking across this globally. So before I move on, just really to say from this part of the talk, it's really to highlight that suboptimal riboflavin status is probably a much bigger issue than we generally recognize, but we just don't have good data globally to back that up. And we became interested in riboflavin, I suppose, we've been interested in it for very many years, and that led us to investigating it in relation to blood pressure and the common polymorphism in MTHFR. So because of the long-standing interest in B vitamins here at Ulster, it really, I suppose, brought us to this very interesting genetic polymorphism. And this polymorphism is C to T polymorphism in the gene encoding for the enzyme MTHFR. And if you'll remember, we need that enzyme in order to generate folate in its active form. And in those who carry the polymorphism, an increased risk of both cardiovascular disease and hypertension is very well recognized within the literature. And the risk is as high as 1.87. So we have studies from, many of these studies are Asian, and some of these are European. And there is certainly a difference, in the risk across the world globally, telling us that environmental factors are also at play. But certainly this polymorphism has been linked with both cardiovascular disease and high blood pressure. So we wanted to investigate this in a cohort of adults from our centre, and indeed we were able to confirm in these patients with premature cardiovascular disease that if you carried the polymorphism known as the TT genotype, your risk of having hypertension, systolic or diastolic blood pressure was significantly greater. And we weren't the first to show that, but we were able to reproduce it. What we were the first to show was that if you administered riboflavin at the dietary dose of 1.6 milligrams per day, you were able to significantly reduce blood pressure. And that was a first. This was a secondary outcome of this study. Homocysteine was our primary outcome. So we were really surprised by this result. And in fact, took quite a long time to get it published, but we didn't give up. And we did indeed get it published eventually in the Journal of Hypertension, because this change in blood pressure was really very clinically significant. We went on to repeat the study in the same cohort 4 years later, where we switched treatments, and then again in an entirely separate cohort, and again demonstrating a significant reduction in blood pressure. We also looked in an observational way at data in each of these lines. I think there are about 200 people represented altogether, and they wore 24-hour ambulatory blood pressure monitors. And you can see that in those with the TT genotype, or the red line, right throughout this 24-hour period, blood pressure was higher in those with the TT genotype compared to those who did not carry this genetic polymorphism. And this data was very reassuring, I suppose, because unlike clinic blood pressure, ambulatory blood pressure is a very robust measure of blood pressure. And we could see the expected pattern over the 24-hour period for blood pressure, but also this the effect of this polymorphism on blood pressure. We then had the opportunity to look in a cohort of 6,000 adults. So we combined the National Adult Nutrition Survey with another cohort of aging that Joel mentioned at the outset, the TUDA cohort, and I'll be talking about them in a while. And we showed that even from as young as 18 years, if you carry this polymorphism, your risk of having higher blood pressure is increased. And the reason why this is important is because this would never be picked up, because you're well below the antihypertensive cutoffs in Europe or even in the US. But what you do is you enter high blood pressure quicker than your non-TT counterparts. So it's important, if you have this polymorphism, to note that blood pressure your blood pressure could in fact be higher. What we were also able to show uniquely, because we measure riboflavin status, is that if you measured riboflavin status and superimposed that on blood pressure, we were able to show that here, this box here represents those who were deficient in riboflavin. So if you have the TT genotype and you're deficient in riboflavin, then you ended up at 3-fold higher risk of developing hypertension compared to someone who had this polymorphism but who had good riboflavin status. So this was really supporting our earlier RCTs that showed by environmental factors such as an improved riboflavin status could really diminish the risk that's associated with this polymorphism. So it's really a lovely example of a gene-nutrient interaction. It's very hard to get other data to support this because nobody measures riboflavin status, but there was a large study published in China a couple of years ago that enrolled 12,000 adults on the China Health and Nutrition Survey. And what this study showed was that those who had the lowest quintile of riboflavin intake, and that equated to a very low intake of 0.59 milligrams per day, that their risk of developing essential hypertension or new onset hypertension was 26% higher than those who were in Q4, which again would represent quite a low amount of riboflavin. So this we feel is sort of mirroring what happens in those who have the polymorphism because their intake of riboflavin is so low. We don't normally see that in developed country, in Europe or in the US. We don't see levels as low as this. And although there is an adaptation in China for low riboflavin intake because of dietary sources, there still appears to be strong enough to link it with hypertension. I'm not going to go into the mechanisms today, but what we hypothesize is that riboflavin is mediating its role through by restoring MTHFR function, which leads to an increased concentration of 5-methyl-THF, which is involved in the generation of nitric oxide and thus regulating blood pressure. And there is some nice data from Greek studies that have used vascular tissue to show lower concentrations of 5-MTHF in those with the TT genotype. So there is some nice evidence to support that, but we certainly we need more. We're currently completing a randomized controlled trial called the INTERVENE study, which not only looks at the blood pressure response to riboflavin, but also looks at the response of FMD, so flow-mediated dilatation. And this is a 16-week intervention in both adults with the TT and the CC genotype using different doses of riboflavin. And we're measuring flow-mediated dilatation, and that will give us an insight into what's happening at the endothelial level. So that study, we're just finishing the last 8 FMDs, and we're going to be ready to analyze. So we're very excited to see what outcome we get there. We've also taken samples to measure nitric oxide in the INTERVENE study. So again, that will allow us to have better insights into the mechanism. And this study has also allowed us to interact with Rima Rosen, who's at McGill University. And Rima really is the queen of MTHFR. And we're going to send our INTERVENE samples to Rima to measure MTHFR enzyme activity. And we've already had a chance to look in a small subset at baseline, and we show significantly reduced MTHFR enzyme activity in those with the TT genotype. And others have shown that, but what they haven't, I suppose, shown is the impact of riboflavin on that. So we're really going to be interested in that. And we also looked in Rima, with Rima and her colleague Karen, we looked at the association between the enzyme activity and total and MTHR, total protein, and again got a very significant association there. So again, once the INTERVENE study has been completed, we'll be able to complete that and also look at the response to intervention. So that's going to be, again, very exciting. Okay, so moving tack, I know time is marching on. Moving tack, I'm just going to come away from one carbon metabolism and just talk about a nutrient-nutrient interaction. And the dependency of vitamin B6 on riboflavin. So one of the other enzymes that requires a riboflavin cofactor for its activity is the enzyme PPO, or pyridoxine phosphate oxidase. And PPO is required for the generation of the active form of vitamin B6 or PLP. And so if we have low riboflavin status, you could hypothesize that our ability to generate active vitamin B6 is compromised. And in fact, way back in 1998, Helene, my colleague, published a lovely paper in HACN that showed in a tiny cohort of only four elderly adults. It was part of a larger study, and this was just a kind of a subset analysis at the end of the paper, but very intriguingly showed that if you intervened with riboflavin in people who had low baseline riboflavin and vitamin B6 status, that you could significantly improve B6 status. So that was showing us that giving some evidence that PPO activity was increased in response to riboflavin intervention, and that in turn resulted in better vitamin B6 status, so an interdependency of the two B vitamins. We had the opportunity to measure vitamin B6 and ERAC in our large Irish cohort, large Irish adult cohort, a combination of NANS and TUDA. And we showed the expected higher vitamin B6 status in males, younger males, decreasing as you age, and higher than females, but becoming very similar. And for ERAC, remember, higher levels indicate poorer status, and curiously, ERAC status seems to improve as we age. But this allowed us to look at the metabolic interaction, and here we see that in people who have optimal riboflavin status at baseline, that their PLP values were about 22 nanomoles per liter higher than those who were deficient, and that's across all age sex categories, a very similar finding. And this, the situation becomes even worse if you're a TT. So the worst case scenario is if you're a TT and you have deficient riboflavin status, then your PLP concentrations are significantly lower compared to if you're a non-TT who has sufficient riboflavin. So that's suggesting that we need riboflavin. We need an adequate intake of riboflavin to ensure optimal vitamin B6 status. But of course, we need randomized control evidence. So we had the opportunity, we combined data from a number of studies that had measured both riboflavin and vitamin B6 status in response to riboflavin or riboflavin intervention. And what we were able to show that yes, indeed, that there is a significant impact in a randomized controlled trial of giving riboflavin, and this is a much bigger population than was previously reported in the Madigan paper. But what I haven't shown here is that the effect size is really driven by those who have deficient status to begin, which makes obviously sense. So really telling us that these two micronutrients are absolutely appear dependent on each other. Okay, so moving on now then to pregnancy. Riboflavin is important in pregnancy, and this is nice data from Keith Godfrey, who ran a large trial called the NIPPER trial, and they reported plasma riboflavin data and show that throughout pregnancy, from preconception to early pregnancy to late pregnancy, that the prevalence of riboflavin insufficiency increases quite dramatically. And why is this important? Well, I'm really going to look at it today. Obviously, it would be important in relation to blood pressure, and I'm not presenting any blood pressure data today, but we absolutely see that in a large pregnancy cohort that I'm going to mention just here. But in the given the time constraints, I'm just going to highlight for you another role for riboflavin, and that's in relation to iron metabolism. So looking at riboflavin in relation to the prevention of anemia. And a couple of groups have looked at that and have shown that in non-pregnant women that yes, in fact, riboflavin status is associated with lower hemoglobin concentrations and a twofold greater risk of developing anemia. The evidence for vitamin B6 is a little bit less robust, but we investigated in a large cohort of over 2,000 pregnant women from the OptiPreg study. And in these women, 31% of the women had riboflavin deficiency at the 12th gestational week. And this is important because it was despite 64% of them reporting to be taking a riboflavin supplement. So we were interested in looking at the impact on anemia in this cohort. And in fact, what we showed that in this pregnant cohort that riboflavin deficiency was a determinant of hemoglobin concentration and was associated with a higher risk of anemia. And we looked at the interaction with both riboflavin and vitamin B6. And while yes, the prevalence of anemia was slightly higher when you combined both, it was largely driven here by riboflavin. So suggesting that riboflavin is also an important nutrient in the prevention of anemia. In terms of randomized control evidence, we gave riboflavin to these pregnant women right throughout their pregnancy from the 14th gestational week until week 34. And here we see that it was a randomized control trial. We had a significant impact on the riboflavin biomarker, showing that riboflavin status improved in these women. And what we also showed, this looks at hemoglobin concentrations. In the cohort as a whole, we didn't see any effect. But again, when we looked in those who had riboflavin deficiency at baseline, we were able to demonstrate a 1 gram per deciliter increase in hemoglobin. And that's important because that is clinically significant. So again, suggesting an important role for riboflavin. Okay, going on then to talk about the TUDA cohort in older age. The TUDA cohort are a large aging cohort that we've recruited across the island of Ireland with Anne Malloy and Trinity College Dublin. And we have followed these now for a period of about over 10 years. So from the original cohort, we've had a follow-up of a 5-year follow-up, and we've also had a few nested randomized controlled trials. And we really have a wealth of data in this TUDA cohort. The most recent being whole genome sequencing, that's opening up a lot of new doors for us and new areas of investigation. I'm just going to flick through this really quickly, given time, but we looked at two aspects that I'm going to just mention to you today. One was cognitive health. So in this TUDA 5+ cohort, we had 1,000 adults who were followed over a 5-year period, and we looked at cognitive decline in those, and we used a repeated battery assessment, which is a very robust tool for measuring cognitive decline. We actually used a number of measures of cognitive function, but I'm just reporting on the R-bands result. And what we showed within this cohort of adults over a 5-year period, that if you had low PLP or vitamin B6 or low riboflavin, that there was a significantly increased risk of accelerated cognitive decline that we did not see with homocysteine folate or vitamin B12. And that may be due to the status that was compromised in these adults, but showing us, adding to the observational evidence that links these two B vitamins with accelerated cognitive decline in this cohort. We also ran a randomized controlled trial where we gave them a cocktail of B vitamins, and we did this in 120 older adults, and this was in about 130 older adults. And the hypothesis being that B vitamins will help to maintain better cognitive function in older adults over a 2-year period. And what we showed here, the R-bands measures different domains. When we looked at the total result, there was no significant difference. But when we broke it down, we did see a significant effect over a 2-year period in the visuospatial domain of the OR bands. So showing us a beneficial effect of a combined B vitamin supplement for cognitive health in this older adult cohort. We also looked at bone health, and this was a trial. It was a 2-center trial that we ran in conjunction with colleagues down at UCD in Dublin. We had 240 older adults aged over 50. So we had TUDA participants combined with a cohort from Dublin. And we had an active placebo of vitamin D and then a B vitamin treatment group with folic acid, B12, B6, vitamin D, and riboflavin. And we looked at, we measured bone health using DEXA scans. And what we saw after 2 years was that in the group as a whole, there was no difference in the change in bone mineral density in the active placebo group compared with the B vitamin treatment group. But of interest, they both declined in the expected range. But of great interest was that when we looked in those with low vitamin B12 status at baseline, using the B12 marker of serum B12, we saw that there was indeed a significant benefit of this B vitamin supplement, combined B vitamin supplement, in maintaining bone mineral density at the total hip. And when we looked at methylmalonic acid, another marker and perhaps considered a better marker of vitamin B12 status, we saw a benefit at the femoral neck. So again, providing evidence that there are advantages for bone health in relation to B vitamins. So who's at risk of B12 status? The older population are certainly at risk due to atrophic gastritis, but also infants, pregnant, lactating women, and also vegans and vegetarians. And there are many causes in the general population or the older adults of why we get low B12. But of interest here, I suppose, in the wider cohort is vegans and vegetarians. And I suppose the reason I've highlighted this is because as populations think about the move to more healthy diet from sustainable sources, we've got to think of our micronutrient status and preserving micronutrient status. This is some work we did with Club B12, an international group of scientists who are interested in B12, and we showed that when you go, it was a review of the literature that very nicely showed that when you go from being an omnivore to a vegan, your vitamin B12, when you go, vitamin B12 intake from the diet decreases significantly. And this is in contrast to the sustainability of the diet. So basically, B12 status is going in the opposite direction to diet sustainability, moving from vegan to an omnivore diet. And I think going forward, I'm not going to go through, but there is some evidence that certainly some limited evidence that shows us that when populations population cohorts have been examined and looked at the comparison of consuming a plant-based diet compared to meat eaters, it certainly impacts our vitamin B12 status. And this is one that looked at a nice study that looked at replacing dietary animal protein sources with plant-based sources and looking at the status of a range of vitamins and minerals in healthy adults. And this was a very nice, robust study, 12-week randomized controlled diet. And again, we can see a very significant impact when we're consuming a plant-based diet. So we have to think about this. We've got to move towards more sustainable diet, but we need to think about how we're going to preserve some of our vulnerable nutrients. And I think that's really important. The data that we have is actually very limited. So there is a lot of work that can be done in this area. We have some large observational trials, we have modeling trials, but the evidence from randomized controlled trials is lacking in terms of the impact, and we really need to be thinking about that. On the island of Ireland, we've recently, as I mentioned, just received, oh, I'm sorry, I don't know what's happening these. We recently received significant funding from the government, North and South, and from the UK government, 32 million in total, which when divided doesn't actually mean that very much. But we're tasked with investigating the impact of moving to a more sustainable diet. And this is a very large project that consists of academic partners, government partners, and partners from policymakers from Oxford and City University of London. And it's a very exciting project, and it will allow us, sorry, my apologies for this, it just has a mind of its own. It consists of 5 platforms. And I and a colleague at University College Cork are leading the nutrition and health platform across the island of Ireland. And we, within this platform, we are tasked with determining the changes required to the existing food consumption patterns to improve population and planetary health, and then developing strategies. We're working with industry in this project to transition to healthy diets from sustainable sources. So what kind of solutions can we come up with to try and ensure that as we move to this more healthy diet from sustainable sources, that we're not compromising our micronutrient or other nutrient intake to any great effect. Okay. So that brings me nicely to the end, a bit of a whistle stop there for the second part of the presentation. But really, I hope that in the last 50 minutes or so, I have convinced you that having a suboptimal B vitamin status is associated with negative health outcomes, and that's across the life cycle. And it's happening in both low middle income countries as well as high income countries. In relation to the vitamins, riboflavin has a novel role in maintaining healthier blood pressure, while deficiency is associated with developing hypertension. It also has a role in maintaining vitamin B6 status and in the prevention of anemia, and that has implications for pregnant women in both low and high income countries. And B vitamins have positive benefits for both cognitive and bone health. So taken in totality, I think these findings really do have implications for emerging dietary recommendations. The work that I have presented, of course, wasn't done by me. I was certainly involved and led some of the studies, but my close colleagues, Helene McNulty, Sean Strain, Christina Pentieva, we have a really active group of B vitamin researchers here at Ulster, and numerous PhD students who really are the champions of the work over very many years, and have been a pleasure to work with and supervise. And we then have a lot of external collaborations and funders and participants to thank for all of the nice work that we've been able to do. Thanks very much. Thanks, Joe.
Thanks, Mary, for that wonderful review of both your research and the research of others. And I want to thank both you and Helene for kind of resurrecting riboflavin as an important nutrient to study. So there are a couple questions in the Q&A, although I might ask my own at some point. But let's let the Q&A people start out. So the first question is, would you say it is beneficial for all old adults to take B vitamin supplements to ensure optimal intake and status?
Yeah. I think one of the things that I didn't really mention, Joel, was our centers, but we're very interested in the dietary level. So we're not really interested in, high intake of B vitamin supplementation. So the work that we've conducted is pretty much all done around the dietary level, or a level that is more or less achievable through dietary intake plus probably fortified food. So I absolutely think that there are clear benefits. I think it's good to take a multivitamin of B vitamins because, we talked about maintaining one carbon metabolism, and I think that a multivitamin is good unless somebody's already taking one of the B vitamins, for some other purpose. But certainly, I think over time at the dietary level, we see clear benefits of B vitamins, across a number of chronic diseases. And I really do think that if we can prevent the deficiency, or we're taking it in a kind of a preventative way, then it really will have benefits through older life. Yeah.
Thank you. Actually, I'm going to ask a question, Mary. When you were talking about the effect of riboflavin on the anemia that we see in pregnancy, if I'm not mistaken, you ascribed that to some type of influence on iron metabolism. And
Yeah.
it wasn't clear to me why you invoked iron metabolism. Couldn't it be riboflavin's effect on, let's say, B6, which you described elsewhere, because there are B6 dependent anemias as well?
There are. When you look at the literature, Joel, I mean, the problem is because not many people measure riboflavin, there's actually very little evidence available. But when you look through the literature, it's a little bit more convincing for riboflavin than for B6 in terms of the observational data. But there's very little randomized control trial evidence. And if it has been conducted, the studies are very small, and they have tended to be more in non-pregnant cohorts. And so we don't have very much data that we can, base our conclusions on. But certainly there is a role for the two B vitamins. In relation to riboflavin, there are a number of potential mechanisms. It might be to do with reducing iron absorption. It might have, there might be gastrointestinal reasons for how it may be impacting iron metabolism, or it could be through iron handling. But probably the biggest is the release of iron from stores. And so that mechanism might be the one that is probably the most likely to be important. But it's a really interesting area. And because, during pregnancy, women who are advised to take iron, it can often lead to a lot of, adverse side effects and complications, and women really don't like taking iron during pregnancy. So if we could even reduce the level of iron and supplement it with the B vitamins, then that might be very interesting in terms of, helping to improve iron status. So rather than just only taking iron, it would be really nice if we could recommend a lower iron intake and supplemented with riboflavin and B6. But we need more data.
Okay, thank you. Let's see. Next question is from my colleague Zhangdong Wang. Zhang says, thank you for your interesting presentation, in particular on metabolic interactions between riboflavin and B6. My question is that riboflavin, for example, by supplementation, can affect the composition and function of the gut microbiome, while the gut microbiota can influence riboflavin and B6 absorption and metabolism. So in your ongoing riboflavin intervention studies, are you going to explore the potential relationship between riboflavin, B6 status, and the gut microbiome under these various disease conditions that you're interested in?
Yeah, really, really, really interesting question. And I think people have talked, the contribution of the gut microbiome to riboflavin status could be quite significant. We really know very little about it. Within the TUDA cohort, we have actually, we're currently doing some work on the microbiome. It's obviously a more invasive procedure, but our TUDA cohort, we currently have a study ongoing where we are looking, we're taking fecal samples, and we will be able to access, do some sequencing on the fecal samples and look at interactions, and also some very small preliminary RCT data that we will be able to share. We're mainly looking at it in relation to cognitive health. So I think that's where we'll start. It's obviously more invasive, more difficult to carry out. And what we really, I suppose, what we really want to do is to try and generate some RCT data, because it's difficult to make sense of observational data. There's so much going on. And I think if we could generate some more robust randomized control evidence in relation to microbiome, that would be really helpful. So we do have a small RCT that we will hopefully be able to report in relation to cognitive health in our older adults. Thank
Okay.
you.
I think the last question we'll take, and we might not get around to all the questions, Mary, although I think you can post written answers in the Q&A if you want afterwards. Anyways, the next question is from Paul Jakes, who I think you know well.
Hi, Paul.
Paul says, wonderful presentation demonstrating the importance of continued study on riboflavin. Many years ago, we were able to demonstrate in the Framingham cohort that individuals with both low riboflavin and the MTHFR TT genotype had higher homocysteine levels, but that this interaction was only present among those with low folate status. Have you examined whether the interaction between riboflavin and TT genotype with respect to hypertension is affected by folate status? Oh, and then Paul also says parenthetically, looking forward to seeing you in Null C.
Oh, great. Great stuff. No, thanks, Paul. And that's a really interesting question. We have, we've actually, we've conducted a study where we have intervened with both riboflavin and folic acid to look at the impact on hypertension. And we don't actually see any added benefit of adding folate. And in fact, curiously, we see a nearly negative kind of an impact when you add in the folic acid, which is difficult to understand. It's sort of, it's nearly neutral or not significantly negative, but a tendency to go more that way. So it's an interesting interaction. And certainly I saw there was a paper published recently. I was trying to access US data just in preparation for the talk today and have a look at what's out there. And obviously there isn't any status data, but there is a paper recently published. I think it's a Chinese group who looked at NHANES, and they see an interesting interaction with riboflavin, and I think it was hypertension or else cardiovascular risk. I just can't remember fully. It's a relatively new paper, but they see the interaction is modulated by they look at it in relation to high or low folate status, and they see it only when folate status is high, I think, and riboflavin status is low. Well, don't quote me 100%, but I can put the paper in the chat. So I think in some populations, and probably that may have something to do with fortification, that the associations are certainly different. So the interaction, there is absolutely a close interaction between folate and riboflavin, and I think that's where we'll see differences in the European versus the US population. So we need to look at our RCT data a little bit closer in relation to the folic acid and the riboflavin, but it certainly doesn't give any huge added benefit. And if anything, it's negative or neutral. But they certainly interact to have different effects.
Okay, great. Okay, I'm going to squeeze in one final question, although we still aren't going to get to all of them. Thank you for your great and insightful presentation. My question is, in your initial slides, you mentioned the prevalence of riboflavin deficiency across China, US, and the UK. Is there any existing evidence showing differences in riboflavin deficiency among different ethnic groups within these populations?
Yes, and that's really down to what they're consuming. So dietary patterns are obviously very different in different ethnic groups, and that has an impact then on riboflavin status. And so I can't quote any data to you, but there are some papers, certainly from an intake point of view, that you can find. The data is there, and it's really down to what you're eating, because in Asian populations, they consume very little milk and dairy products because of lactose, high prevalence of lactose intolerance. And so for that reason, they're dependent on plant sources of riboflavin, which do supply riboflavin, but obviously in much lower concentrations. And there is a urinary adaptation reported within Asian populations. So perhaps their requirement, may not be as high. But certainly, if you look even within the UBC population that we measured, because there was a high Asian percentage of adults within that cohort. And I think dietary intake sources were quite different, and that impacts your intake. We obviously, and also the status in that particular group. You can see it really clearly, even if we look in our Irish population. If you look at milk consumers versus non-milk consumers, it has a really significant effect on riboflavin intake. So again, as we transition to more sustainable diets, we need to think about where we're going to get our nutrients and our micronutrients from. And I think that's really important. We don't have good data to back that up. A lot of the data on sustainability is based on modeling. Some of the modeling is excellent, but we need good, human studies and RCTs to show us the impact of moving towards more plant-based diets. You certainly can do it, but at population levels, we really need to be careful.
Okay, I think I'll stop there. We're past the hour as it is. Mary, thanks again for a terrific talk, and most importantly for this wonderful research that your group continues to generate. Okay, take care, and there's one or two more questions in the Q&A if you want to try to address those. Okay,
That's
take
great.
care.
Thanks very much, everybody. Lovely to see everybody, and I'm looking forward to seeing anybody I know in Nancy at the end of June. Thank you.